Hello friends, now let's start with the one term that is called a biotechnology. When we talk about the biotechnology, what comes in our mind? The bio means nothing but a life. Anything which related to life is called the bio. When you talk about the technology, that has to be something related to technology or related to life. So this is the exact meaning. So let's talk about what is the definition of biotechnology. We can say biotechnology is nothing but a technique by which there is a alteration in the genetic makeup of the bacteria for the human welfare. So there are two words that we have to understand. There is a changes in genetic makeup of bacteria, or we can say it is nothing but a hybridization. Okay, and what reason this is biotechnology is done? For the human welfare. Definitely, whatever I'm going to do is that human welfare. So, what is the definition? I want to repeat it again. Biotechnology is a technique in which there are the brings about some changes in a genetic makeup of the bacteria and hybridization for the human welfare. Let's talk about the example and let's clear it about insulin. Okay, so the example is insulin. Let's study what is the example and what is the depth of the biotechnology. Okay, study that the example is insulin. So now tell me what is the insulin? The insulin is secreted by beta cell of pancreas, or in more specific manner beta cell of Langerhans of the pancreas. So we all know that inside the cell there is a nucleus and there is a DNA. What is the DNA molecule? DNA is nothing but a genetic factor which is responsible for all the activities, right? So when you talk about when this is removed outside, if this is a DNA is the longest one, we know that it is made up of the sequence. That is, we know that A, T, G, C, adenine, thymine, guanine, cytosine. So now there is a some sequence responsible for the some secretion of the enzymes and the hormones. So if you take a fragment or the segmented things fragment from the human DNA, then if suppose let's take example of the insulin, we are talking about the insulin, let's take example of the same. If the segment on the DNA is responsible for the secretion of the insulin, if I take that fragment away from the human gene, then this look like this. Suppose this is the fragment which is responsible for the insulin secretion. Now let's talk about the E. coli. E. coli is full form Escherichia coli, which is a very nice bacteria. Now let's talk about this. Suppose this is a cell inside the E. coli. It has its own circular nucleus, or the DNA we can say. Now tell me one thing, what is the genetic makeup? Change in the genetic makeup, that means that suppose this is the DNA is responsible for the insulin secretion, we can change over the E. coli DNA by adding this fragment here. This is very important thing. So now it will be changed like this. Suppose that this is, we have to break it open, right? So now the circular DNA now going to change like this. So this part is nothing but a human genes which is joined to the present E. coli DNA. Now what is this E. coli do? It reproduce again and again, and then while reproducing, while making a multiple copies, this responsible for the insulin secretion. Now as the application or applied the biotechnology, what going to happen is E. coli is not aware about that the DNA has been changed, but now we know that if this is the fragment responsible for insulin secretion, while multiplying or while taking on nutritions, this DNA also get copied. There are 1,000 copies of the E. coli will be produced, and this 1,000 copies of E. coli will secrete by nature only, because now changes, genetic makeup has been changed, hybridization has been occurred. So now they will secrete the insulin. So now what we have done is we done that we changes the E. coli DNA into, not exactly we have changed, we have just incorporate the human gene which is responsible for a factor responsible for the insulin secretion, we have to incorporate into the E. coli DNA and we get the result as a it will be insulin secretion. So in nutshell, biotechnology is like this. So there is a change in genetic makeup for the human welfare. Earlier insulin prices are very high, but because of this technology, we are getting tremendous amount of the insulin, so price has been low. And this is biotechnology is a very booming field, you can say. There are many changes. We are using the bio, that life or living organisms, for the human welfare. So I hope you understand this very well. Thank you.